So, microbial metabolism, or just metabolism in general, is the sum total of all chemical reactions within a cell. And when we're talking about metabolism, we have to start with thinking about this molecule. This is adenosine. Adenosine, or adenine, is a nucleotide. This is actually the A that binds with thymine in our nucleic acids. But if we were to stick another phosphate on that, we would call that adenosine diphosphate, or ADP. Now, sticking a phosphate on there at the end, that's a pretty high energy bond. That bond doesn't like to stay on there. And if we were to stick yet another one on there, that would be adenosine triphosphate, or ATP. And in order to force that last phosphate onto that particular molecule, it requires a lot of energy input. And that's why ATP is the currency of energy within a cell, because that particular bond contains a lot of energy, where if that phosphate were released and you turn the molecule back into ADP, adenosine diphosphate, you would release a lot of energy too. So when we're talking about metabolism, we're talking about the sum total of all reactions in a cell. We're talking about catabolic and anabolic reactions. Catabolic reactions are where we break down molecules, start with large molecules, and turn them into smaller ones. And every time you break down a molecule and you break those chemical bonds, you release a little bit of energy. You can take that little bit of energy and use it to force that last phosphate onto ADP and make ATP. Catabolic reactions, you can remember, are the large to small, releasing energy. My little trick for remembering that is cats destroy everything. So you can start out with nice curtains and a nice couch, and those cats are going to burn all kinds of energy by shredding your curtains and your couch into tiny bits of fabric and fluff. Anabolic reactions are the building up of larger molecules. So think about our macromolecules, where we have to make nucleic acids, we have to make protein. Bacteria have to make peptidoglycan, or phospholipids have to be synthesized. All of these things have to start with individual monomers and small molecules. And so anabolic reactions are the small to large. And in order to form something, so think about, you're building a brick wall. You start out with tiny bricks, and you have to add energy. It takes a lot of energy to build a brick wall, to make something large from something small. So this is why ATP is actually considered the currency of energy within a cell, because it is the one that shuttles energy back and forth between our catabolic reactions and our anabolic reactions. I like the way that this particular picture represents that, because it is a cycle. It does go back and forth. Catabolic reactions are going to release energy. And so when we're talking about releasing energy, we have to think about, we have to talk about a couple words that might be new, and that's exergonic and endergonic. So when we're talking about energy and catabolic reactions, where energy is released, do you think that catabolic reactions are exergonic or endergonic? Of course, they're going to be exergonic, right? Because that's releasing energy, exer out. And so that energy is trapped in ATP. We use that energy to push that last phosphate onto ADP and make ATP. You could even consider the formation of ATP to be endergonic, because it requires an input of energy to push that phosphate on. And then when you take ATP over to anabolic reactions, breaking down ATP to ADP, that's exergonic, that releases energy. Anabolic reactions are endergonic, because they require an input of energy. So you could actually resketch this. That's one of our guide objectives, is to sketch this, add the terms exergonic and endergonic where they're appropriate. Okay, so you know this one. Catabolism is a form of metabolism in which blank molecules are converted into large molecules are converted into small. That's correct. Okay, this one always gets students, so I want you to actually pause your player and really think about this. This is asking you a reaction that requires ATP to be broken down into ADP is a blank reaction. So I'm not asking you about ATP to ADP. I'm asking you about the reaction that it's coupled with, the reaction that requires ATP to be broken down. Okay, pause your player. Okay, here it comes. ATP being required to be broken down into ADP, that means that ATP has to release energy, which means that if it's required, this has to be an endergonic reaction that requires that to happen, so that that energy can be transferred to an endergonic reaction, and anabolic reactions are endergonic reactions. Okay, when we're talking about energy and macromolecules, carbon in particular, we have to talk about our trophic groups. So there are two major types of trophic groups and then two subtypes. So there are organisms that can make their own organic carbon from carbon dioxide. So those that can make sugar, basically, from carbon dioxide, those are called autotrophs. And then there are heterotrophs. Those are ones that have to take in sugar from outside their body or outside their cell. We're heterotrophs, right? We have to eat food. Then if we talk about either photo or chemo, we're talking about where does that energy come from. And so if you're asking phototrophs, auto or hetero, phototrophs, where do you suppose they get their energy from? Photons, right? Photons come from sunlight or light, so these guys are going to be able to get their energy by using photons from sunlight. Chemotrophs, either chemoauto or chemohetero, chemotrophs get their energy from breaking chemical bonds. When you break a chemical bond, it releases energy. So think about, first question, where do they get their organic carbon? Do they make it themselves or do they get it from outside their cell or body? That's going to tell you whether they are auto or heterotrophs. And then you ask, where do they get their energy from? Do they get it from light or do they get it from breaking chemical bonds? And that will tell you the difference between phototrophs and chemotrophs. So let's talk about each one of those groups just briefly. Photoautotrophs. What's a photoautotroph? Where do they get their carbon from? They make it themselves from carbon dioxide. Where do they get their energy from? Sunlight, right? So these are our photosynthesizers. These guys are going to use the energy from sunlight to turn carbon dioxide into sugar and other things that they can use within the cell. So they still require sugar, but they actually are going to build that sugar themselves using energy from sunlight, right? So photosynthesis is actually an anabolic reaction, right? Because you're taking carbon dioxide, which is a very small carbon molecule, and you're making things like glucose, which has 6 carbons. So we require an input of energy. And where does that energy come from? It comes from sunlight. We actually make ATP from sunlight. Now, when you're talking about photosynthesis, that can actually be oxygenic or anoxygenic. What that means is it can make oxygen in the process as like a waste product, or it cannot. And it depends on what type of light-absorbing molecule they use in the process by which they make ATP from sunlight. Next up is chemoautotrophs. So where do they get their organic carbon? They're going to make it themselves. And where do they get their energy? It's not going to be from sunlight this time. It's going to be from breaking chemical bonds. Okay, so these guys are going to make their own sugar molecules, or they're going to make their own organic molecules, but they're not going to use it, or they're not going to do it by sunlight. Instead, they're going to take inorganic molecules and they're going to pull electrons off them and stick them onto simple things like hydrogen and carbon dioxide. They're going to use the energy from pulling electrons to fuel their cellular reactions, and they'll probably also use the simple molecules that they made. These guys are going to be ones that are found in really extreme climates where sunlight isn't available, like deep sea vents. So where do you suppose you might find representatives of this chemoautotroph group? Right, you got it. Deep sea vents, that was your clue, right? So think about if they don't require sunlight, and they're going to take electrons and they're going to stick them onto things like carbon dioxide and hydrogen, they're actually going to make things like methane in that process. And remember, our methanogens, those are also going to be our archaea. Next up is chemoheterotrophs. Okay, so chemoheterotrophs, where do they get their organic carbon? Are they going to make it themselves? Or are you going to get it outside their body? Outside their body, hetero, outside. And how are they going to get their energy? They're going to get it from breaking chemical bonds. So we are chemoheterotrophs. Now, what we do is we take organic molecules and we break them down, things like glucose. We break them down and we release energy, and we use that energy trapped in ATP to fuel our anabolic reactions. Now, not all chemoheterotrophs fall into these two major groups, but there are two large groups of chemoheterotrophs, two ways of bringing in that organic carbon. There are saprobes and there are parasites. So saprobes, I already talked about them a little bit when we were talking about fungi. Fungi are saprobes. These guys often are important because they can't just bring that organic molecule into the cell. If it's too big, they have to excrete extracellular enzymes in order to do that. They're the ones that are going to feed on dead organic things. That's what makes them saprobes. Sometimes they can actually cross over and become parasitic if a host is susceptible, so that might make them opportunistic pathogens. And my favorite example of these guys are our gram-negative bacilli that are soil-dwelling saprobes. They produce a pigment called pyocyanin, which is blue, and another pigment which is called pyoverdine, which is green. This is Pseudomonas aeruginosa. This is the one that smells like artificial grapes. Remember, these guys are saprobes, so they feed on dead organic molecules. They can become opportunistic pathogens of people like burn victims. So this poor person has had some severe burning, and somehow this Pseudomonas aeruginosa has gotten established on this dead skin, eating the dead skin, but unfortunately, once they get established, they can start parasitizing living tissue. So if you were ever in a burn ward and you saw this blue-green, or you were to smell artificial grapes, that would be a big warning sign of a Pseudomonas aeruginosa infection. Fun little side story about this particular guy. When I had students swabbing their environmental samples in lab at the last place I was at, One student had Pseudomonas aeruginosa in one of the 4 quadrants on the plate. They got to swab 4 things. And I was so excited about it. I showed everybody and I told them all about it. And then I turned to her and I said, "What did you swab? Where did this come from?" And she said, "I swabbed my surgical site." So she didn't have like some sort of infection or anything, but they were there. She had a ankle surgery or something like that, and there was a little bit of dead skin, and from the incision, and they were happily munching away at that dead skin. Okay, next up, parasites. All right, so parasites are going to be the ones that live on living tissue, and just by the act of living on living tissue means that they're going to cause damage. That's why they're pathogens. That's why they're parasites, so they cause disease. Now, how much damage they cause is dictated by just how much carbon, how much nutrients they need, and that they can't make themselves. So there are some obligate parasites that are unable to grow outside the cell, things like leprosy and syphilis. So our Nocardia species and Treponema pallidum. Others we can grow in culture, like Neisseria gonorrhea and Streptococcus pneumoniae, although both of these organisms are something that we call fastidious. Gonorrhea requires us to grow it on boiled blood agar called chocolate agar, because it needs 2 growth factors that it can't produce itself that can only be found inside red blood cells. Streptococcus pneumoniae can break down blood on its own, so as long as we grow it on sheep's blood agar with fresh blood, it can grow, but we can't grow it on a regular TSA plate like we do most of our other microbes in the lab. Then we even have obligate intracellular parasites. Things like rickettsias and chlamydias, things that actually have to live inside human cells in order to survive. Last group is photoheterotrophs. Photoheterotrophs, so where do they get their organic carbon? They get it from outside their cell. Where do they get their energy? Get their energy from sunlight, not from breaking down chemical bonds. This is kind of a weird group. So they're going to use sunlight for energy. So they're going to use sunlight to make ATP for their anabolic reactions, but they take their organic carbon from sources other than carbon dioxide. So they don't synthesize their own organic carbon. They take it from outside their cell. And because they're such a weird group, I actually had a really hard time finding examples. These guys would be our purple and green non-sulfur bacteria. Purple and green non-sulfur bacteria. So these are going to probably be organisms in the Archaea group again. Although, oh gosh, if you look over here in the right where it says B, it says Rhodospirillum rubrum, and that's actually one of the bacteria that we use in lab. We've looked at that guy. So purple and green non-sulfur, but Rhodospirillum rubrum we know is red. So interesting, yeah. Okay, I put this slide in here. Normally this would be kind of a nice little breaker in the middle of lecture where I just talked about all the categories. We would list them, and then I would pass out the markers and ask you to fill in the blanks with what the names of the categories and the energy source. So start out with, right, we've got the 2 pink rows. Start out with where do they get their organic carbon? Are they hetero or auto? And then the orange categories are going to be, are they chemo or photo? Where do they get their energy? And then remember we had 2 major groups of our chemoheterotrophs. Not all chemoheterotrophs fell into saprobes and parasites, but they did. And then think of microbe examples. Try and think of the examples that I told you just now. Go back through the slides, and then try also to think of your own examples that would be microbial. All right, Metabolism is the sum total of catabolism, photoautotrophs, ultimate source of energy. I'm going to let you go ahead and slide around on your player, pause this, see if you can fill it in, pause this one, see if you can fill it in. And then I'm going to end the slides here. You can't ask me questions. That's the unfortunate part of a video. That's why lecture is so good. So that you can do the guides ahead and you can ask me questions when we're together and we can practice. And I'm going to move on to the next topic in the next video. This is a good break spot. So on to the next one.